Hello, my name is Maria Stephenson, and I'm a co-op student at the Dr. Nashville Lab at Canada's Michael Smith Genome Sciences Centre. This is my poster for RNA-Scoop, a tool which provides an interactive visualization of isoforms and single-cell transcriptomes. Single-cell sequencing has become a powerful tool, allowing researchers to examine transcriptome heterogeneity at the single-cell level. However, analysis of single-cell RNA-seq data is typically based on gene expression, ignoring information at the isoform level. Each gene may generate multiple transcript isoforms through alternative splicing events and alternative transcription start and termination sites. Because of this, a gene may have multiple functions, and studying isoforms is crucial to understanding the functionality of the genome. For this reason, we created RNA-Scoop, to allow researchers to easily visualize isoform expression across thousands of cells. The input of RNA-Scoop is a single JSON file, which specifies paths to a GTF file containing the isoforms of interest, a matrix file containing their expression levels in each cell, and files containing labels for the matrix rows and columns. Users can select genes for the isoform view, where all isoforms of selected genes are displayed. To do this, users open up the Gene Selector dialog. Users can search for genes by ID or name, or scroll through the genes table to find genes of interest. The table is sortable by ID, name, number of isoforms, and maximum fold change across clusters in the t-SNE plot. The ability to sort genes based on maximum fold change is important because it allows users to easily identify differentially expressed genes. In the isoform view, the isoform representation is similar to that used in UCSC Genome Browser and IGV. Exons are represented by boxes, and lines represent spliced introns. The isoforms are colored according to their average expression in the cells in the t-SNE plot. Now it's important to note that the isoforms are selectable. When a user selects a group of isoforms, the cells which express those isoforms are highlighted. Additionally, a dot plot is drawn next to each isoform, allowing users to easily compare expression between clusters. The dots are colored according to the isoform's average expression in the cluster, and the size of each dot represents the percentage of cells in the cluster which express the isoform. For example, looking at this dot plot, we can see that the H3F3AOS isoform is expressed across all clusters in the t-SNE plot, and that the different isoforms of the APOE gene have very different expression patterns, with the 7th isoform being expressed in 7 out of the 10 clusters, and the first isoform being expressed only by a small percentage of cells in the 6th cluster. Now, by default, isoforms are drawn with respect to their exon's chromosomal coordinates, as if the corresponding genes were on the positive strand of the genome. However, users can choose to reverse complement isoforms on the negative strand, so that they are drawn in their 5' to 3' orientation. This can be useful for datasets produced by strand-specific sequencing and in cases where gene orientation is known. Additionally, in order to hide potentially incomplete isoforms and misassemblies, we give users the option to hide single exon isoforms. Users can adjust the gradient by which isoforms and dots are colored by opening the TPM gradient dialog. Users can define their own range of TPM cutoffs, change the colors of the gradient, and transform the gradient from linear to logarithmic scale, and vice versa. The t-SNE plot is completely interactive, allowing users to zoom in and out of different areas and pan the plot freely. Cells are selectable via lasso selection. When a user selects a group of cells, the isoforms are colored according to their average expression in the selected cells only, and the dot plot is generated from the selected cells only as well. It is important to note that the t-SNE plot also supports multiple label sets. Users can switch between label sets using the Label Set Manager. This dialog also allows users to create new label sets via selection of cells in the t-SNE plot. New label sets can be saved and used at any time. So, in summary, isoform detection and discovery in single cells are central to advancing our understanding of cell diversity. Through use of an interactive t-SNE plot, RNA-Scoop allows users to easily explore isoforms in a single-cell transcriptome dataset of thousands of cells. This project has been funded by Genome Canada, Genome British Columbia, and the National Institutes of Health, and my funding for this conference was provided by ISCB through their fellowship award. If you would like to use RNA-Scoop, it's available on GitHub. Just follow the link or use the QR code. Thank you for listening, and I hope you enjoyed my presentation.